moving on to the next topic, which is anticoagulant agents. Let's talk about anticoagulants. Before talking about anticoagulant agents and antiplatelets and fibrinolytics, we'll give a basic introduction about how normally a clot is being formed. If you could understand that basic concepts, we can just find out different spots where we can inhibit those particular areas. Again, this will not be the reference for pathology or physiology body. That will be dealt by individual faculties. So here I'm just going to be targeting only those areas which is there in patho and physio, which is clinically important. For example, for which we have designed drugs, only those clinically important areas will be targeting more. So let's start with the first step, and that first step is platelet activation. We have two steps to be explained. One is called as platelet activation, the other one is platelet accumulation. We'll be talking about platelet activation at first. Consider here is where your blood vessel is. The blood vessel is getting broken down, and that's what bleeding is going to be happening. We all know that. Whenever the blood vessel is getting broken down, as we know, the bleeding will start to occur. When the bleeding occurs, we have the first platelet which comes into contact with the collagen, gets activated, thereby releases out a substance which will bind with its own receptor which is there on the other platelets. This will bind with its own receptor on the other platelets, thereby it will produce something called as activation cascade. Simple concept, the platelet is calling out for help. It is just informing to every individual platelets that there is some issue which is happening over here. We have a bleeding which is there. Please come towards this spot. We have to treat this particular patient. Now we're going to be looking into this particular complex body. We're going to be looking into how exactly this happens, what exactly is released from the platelets, and how it is inducing the activation of the other platelets. I'll be explaining about that in detail over here. Consider here is where the single, that second platelet which we have. On the second platelet, there will be multiple different receptors, guys. We have the first receptor to be explained, 1, 2, 3, 4, 5, 6, 7. If it is passing through the membrane, we have discussed that in general pharmacology. It is a G protein coupled receptor because it is like a longer receptor. When we compress it, it passes through the membrane 7 times. It is also called a 7-pass transmembrane receptor. Now this is actually a GI coupled receptor. Similarly, there will be one more, 1, 2, 3, 4, 5, 6, 7, which is GS coupled receptor. Two different receptors are there on the surface of platelets body. These are the platelets. Now what exactly happens? Now, as we told, this is the previous platelet. From the previous platelet, there will be release of a substance which is named as ADP molecules, adenosine diphosphate molecules. This ADP in turn will bind with the GI receptor which is there on the surface of the platelets. We have discussed about that already in general pharmacology itself body. And this is what type of receptor? P2Y12 receptor. This is P2Y12 receptor. When ADP comes and binds with it, it's going to be stimulating it. On the other hand, what exactly is the one which is stimulating GS receptor? Here there will be adenosine. We know adenosine will be binding up with this GS receptor, which is actually A2 receptor, which is actually A2. Adenosine has got A1, A2A, A2B, and A3. Out of this, A2 receptor is there over the platelets, which is actually a GS coupled receptor. Whereas A1 and A3 are GI coupled. We have discussed all of these points in general pharmacology itself. Now, when adenosine is coming and binding with this receptor, it is a GS, whereas ADP, adenosine diphosphate, when it's coming and binding with it, which is GI, which means what is the receivable concept? The receivable concept is going to be adenylyl cyclase. Because we know the difference between GS and GI, guys. GS receptor will be stimulating adenylyl cyclase, whereas GI receptor will be inhibiting adenylyl cyclase. Two opposite responses which is happening. Now try to guess now what exactly happens whenever adenylyl cyclase is getting stimulated. What's the second messenger? Cyclic AMP. This cyclic AMP is responsible for platelet activation. What exactly is the responsible concept? I will explain that in few minutes. See, whenever adenosine is coming and binding with the GS receptor, I'm talking about the right hand side. Adenosine is coming and binding with it. You know what happens? It will stimulate adenylyl cyclase. What do you expect will happen to the levels of cyclic AMP? That is going to be raised. Whenever there is an increase in cyclic AMP, that will produce inhibition of platelet activation, that will have inhibitory activity on platelet activation, which means for prevention of clot, I will repeat that statement. For prevention of clot, adenosine is good and ADP is bad. I hope you can understand what I'm saying. I will repeat that statement. For prevention of clot, adenosine is good and ADP is bad, because adenosine will prevent the platelet activation, whereas on the other hand, ADP will stimulate platelet activation. And how is it happening? We'll go with the same color. Because it is inhibiting adenylyl cyclase, so thereby cyclic AMP levels will be decreased. When there is no inhibition which is happening, what do we expect? Platelet will be more activated. So here we have an equilibrium, guys. We have an equilibrium between ADP and adenosine. ADP will be inducing platelet activation, whereas adenosine will be inhibiting platelet activation. This will be there in equilibrium. That's the reason why usually these platelets will not be producing any clot. But unfortunately, we have one small issue. That issue is, in terms of a breakdown, whenever the blood vessels are getting broken down, there is extraordinary release of ADP. So one point which is going more favor for ADP. The second point is adenosine is there now. This adenosine, we have the blood vessel over here. We have the blood vessel. Inside the blood vessels, we have RBCs. RBCs. The RBCs needs adenosine. It has a separate transporter on the surface which is responsible for adenosine to be reuptook back inside the RBCs. It is called as adenosine uptake by the RBCs. So adenosine is prevented to be accumulated there near the platelets. So 2 points are happening over here. The first one is what? Shall we give a small revision of what we have explained? ADP is inducing clot, adenosine is preventing the clot. One point which is going in favor of ADP, too much accumulation of ADP. So there is more ADPs which is there moving towards clot formation. And number two, adenosine is there now, that is not allowed to be producing its mechanism for a longer duration. Instead, adenosine will be back inside the RBCs. It will be taken up inside the RBCs. So there is no one to take control of ADP. Too much ADP levels which will stimulate the platelet activation from happening. Is that clear? Okay, that's how platelet activation is going to be spread out. Again, from this platelet which we have drawn, there will be release of ADP, which will bind with the other platelets. Those platelets will be moving towards that, and those platelets in turn will be releasing out those molecules. This is what we explain as an activation cascade. Platelet activation cascade. We have explained that from the knowledge which we have gained from general pharmacology itself. Now moving on to the second step, which is called as platelet accumulation. The second step is called as platelet accumulation. Again, here we have the blood vessels which are getting broken down, and we have the first platelet which is accumulated there, or platelet which is moving on towards that. As I've told, platelets are getting activated. It will start releasing out a substance. That will be releasing off a lot of different substances. I told about one substance previously, which is ADP. Not only ADP, guys, multiple different substances will be released. This again will be binding with other platelets. On the surface, we have receptors, and this will bind with those platelets, tend to produce accumulation. Here, we're not talking about ADP, we're talking about one more molecule, which is called as Thromboxane A2. I'm going to be explaining about what exactly is happening at this point. There will be release of Thromboxane A2 from platelets. So what happens, buddy? Whenever Thromboxane A2 is getting released, it will bind with its own receptor which is there on the other platelets. So Thromboxane A2 will bind with Thromboxane A2 receptor on the other platelets. So just imagine this. This is what the first platelet is, and this is what the second platelet is. Good. From this platelet, there will be release of Thromboxane A2, which will bind with the Thromboxane A2 receptors of the other platelets. Now, when this thromboxane A2 receptor is getting stimulated on the other platelets, there will be an interlink which is happening between these two different platelets, which will start expressing out one more receptor from the surface. There will be receptor, there will be expression of, as we can see, my little figure is getting popped out. There will be expression of one more receptor which is expressed out from the surface. This receptor is called as GP2B3A receptor. But as we can see here in this, in my hand itself, this thromboxane A2 thromboxane A2 receptor are binding up with, but GP2B3A are not binding up with each other. We need an interlink, we need a bridge to be formed between the glycoprotein 2B3A receptors, so that the link is going to be formed, and that bridge is formed by fibrinogen. We'll explain that. So what happens, because of the interaction between thromboxane A2 and thromboxane A2 receptor, there will be expression of glycoprotein 2B3A receptor. This glycoprotein 2B3A receptor which is getting expressed out, I will draw the diagram also over here, my dears. Here is where the platelet is, and here is where one more platelet is going to be, from which there will be release. I'm going to be drawing it like this, which is thromboxane A2, which will bind with thromboxane A2 receptor, because of which there will be expression of GP2B3A receptors, glycoprotein 2B3A receptors. And between this glycoprotein 2B3A receptor is where the bridge is going to be formed, and that bridge will be formed by fibrinogen molecules. Which means here the expression of GP2B3A will interact fibrinogen molecules. This fibrinogen is going to be there at inactive form, or to say it is a precursor form. We have to convert it to the active fibrin strands. Fibrinogen in turn will be converted to the active fibrin strands with the help of, as simple as that, clotting factors. With the help of clotting factors, this fibrinogen is getting converted to fibrin. So what exactly are those clotting factors designed or derived from? By two different pathways. One is called as the intrinsic pathway. The other one is extrinsic pathway. Most of you would be knowing that these intrinsic and extrinsic pathways are important long answers in your first year physiology. But what exactly is the function of this intrinsic and extrinsic? To produce thrombin, no? Factor 2A has to be synthesized, which is responsible for conversion of fibrinogen to fibrin. This is how the clot is getting formed, guys. As we can see, this is how the image looks like between two different platelets. This is the first platelet, and this is the second platelet. Similarly, there will be multiple different platelets. For example, there will be one more platelet. Again, here there will be expression of GP2B3A, GP2B3A. There will be interlink with the help of fibrinogen molecules, which will again be converted to fibrin strands. Again, there will be one more platelet, from which there will be expression of GP2B3A, and fibrinogen will be forming that link between those. Thereby, there will be a complete linkage of all the platelets, which is just preventing the bleeding from happening over there. But consider this blood vessel is getting broken down, right? And that clot is getting formed with platelets are there and thromboxane A2, thereby GP2B3A, which is interlinked with the fibrin strands. Now we have converted it to fibrin strands, as simple as that. We have the clot which is formed, which is preventing the bleeding from occurring. We have the blood vessels which are getting broken down. We'll wait till the blood vessel cells, that smooth muscle cells is getting proliferated. And once this blood vessel is getting fused up, there is no need for the clot, right? Now what happens is, just imagine this is what fibrin strand is. This is what a fibrin strand is. Every fibrin strand will be attached with the molecule called as plasminogen molecule. Every fibrin strand, even if this fibrin strand also will be attached with one more molecule called as, there will be one more molecule which is attached, which is called as plasminogen molecules. Every fibrin strand will be attached with the plasminogen. These are called as fibrin bound plasminogen molecules. Now, what happens over here is when the blood vessel cells are getting proliferated, the endothelial cells are getting fused. From the endothelial, there will be a release of a particular molecule. These molecules are going to be activating the plasminogen. So, as you remember, this is what your fibrin strand is. On the surface, we have plasminogen molecule. Whenever there is release of this plasmin, that molecule, which is going to be activating it, that is called as tissue plasminogen activator. Why? Because it is released from the endothelial tissue. It will bind with the plasminogen and tend to activate it. Once it is getting activated, it is going to be cutting out these fibrin strands. So this is what inactive plasminogen is, and this is what an active plasmin is. So all the plasminogen will be cutting up this fibrin strands. Whenever the fibrin strands are getting broken down, the platelets are getting dislodged. It will just mix up with the blood. This is how the clot is getting dislodged. We'll be explaining about that. We'll be writing it, don't worry. This fibrin strand will be broken down to inactive segments with the help of a molecule, and that molecule is plasmin. You remember that? Which is called as plasmin? Is this or this? Yes, this is what called as plasmin, which is synthesized from plasminogen molecules. So plasminogen, which is a precursor, will be converted to the active plasmin, and this conversion will happen with the help of Plasminogen Activator PA, which is released from the endothelial tissue. Vascular endothelial tissue. That's why this is also called as tissue plasminogen activator. Is it understood, buddy? Well, that's it about the basics of how clot is getting formed and how the clot is getting broken down. Now, I hope if you are logically thinking, you would have come across this doubt. Even if you have not come across this doubt, don't think that you are completely illogical person. Don't think that. No. I'm here to explain. Don't worry. Consider here is where the blood vessel is. Blood vessel is getting broken down, and here is where the clot is. Good. When the blood vessel is getting proliferated, as we can see, even before the blood vessel is completely being fused, we have a part of endothelium which is there inside the clot itself. What if even before the blood vessel is getting fused, this from the endothelium, a tissue plasminogen activator is getting released, that is activating plasminogen to plasmin, thereby it is breaking down all the fibrin strands, thereby the clot is getting dislodged, again the patient will start bleeding again. To prevent this premature activation of plasminogen to plasmin to occur, we have a physiological molecule which will inhibit the tissue plasminogen activator itself, and that is called as the physiological molecule which will inhibit the tissue plasminogen activator. Just give me an example. What would be its name? As simple as that. Plasminogen Activator Inhibitor, called as PI. Plasminogen Activator Inhibitor is the one which inhibits tissue plasminogen activator. This is very much important clinically, guys. Why? Because we'll be designing certain drugs which are actually structural analogs of TP. Okay. But unfortunately, we have a negative point that structural analogs of TPA would also be broken down by pi. That's the reason why we have to think of this. This is a very important clinical implication to be remembered. This is how the whole clot, how platelet is getting activated, how platelets are getting accumulated.